Okay, hi. So my name is Julien Gagneur. I'm going to chair the next two talks. And our first talk is in oncology. This is going to be by Mike Berger. Mike did his PhD at Harvard University and his postdoc at the Broad Institute. And he's now Associate Director of the Marie-José and Henry Kravis Center for Molecular Oncology at the Memorial Sloan Kettering. His research is focusing on developing and applying methods for profiling tumor DNA to characterize the genetic makeup of individual cancers. Michael's talk today is entitled Building a Scalable Genomics Program, How Tumor, Normal, and Plasma DNA Sequencing Are Informing Cancer Rare, Cancer Risk, and Cancer Detection. So Mike, thank you. The stage is yours.
Thank you. Before you disappear, can you confirm that this is displaying correctly? Does it look okay?
Yes, it's good.
Okay, good. Thank you. Well, thanks so much. And thank you for the invitation. We'll test the strength of the internet signal from New York City now. So it's as you heard, this is going to be a cancer-focused presentation. I work at Memorial Sloan Kettering Cancer Center, which is one of the largest cancer centers in the United States and actually around the world as well. And we have invested pretty heavily over the last 8 years or so in a precision oncology program. Oh, I apologize, my disclosures are here. And the sort of cornerstone for that program has been a test called MSK Impact, and I'll be presenting a lot about the data generated through this MSK Impact test. It's the clinical workflow is shown here, starting with patients consented by their primary oncologist in the clinic, and I'll come back to this. Usually I gloss over that part for many of the talks that I give, but I think it's worth spending a minute or two discussing that aspect. We collect DNA from both the patient's tumor as well as matched normal tissue, usually blood for patients with solid tumors, prepare separate sequencing libraries, and capture using probes, now capturing 505 genes that as a cancer center we've identified as relevant for either clinical oncology or frequently mutated from the cancer genomic studies that have been published already. Sequence deeply to about 700 to 800x coverage, so that gives us high sensitivity as well as precision for identifying the specific variant allele fractions, which help us interpret not just the presence or absence of mutations, but also the clonality and the zygosity of mutations as they appear in the tumor. Several parallel bioinformatics analysis to identify many different classes of genomic alterations, including mutations, indels, copy number alterations, gene fusions, signatures like microsatellite instability and tumor mutation burden, which have emerged as important biomarkers for immunotherapy, as well as other classes of variants that I'll talk about. And this is analyzed, interpreted, reported by an integrated team of bioinformaticians and pathologists to produce sort of the final set of alterations that we've aggregated into a database, annotate using a knowledge base called ONCOK that I'll also speak a little bit about. And this is used to produce individual clinical reports, but also enable some of the larger scale data mining research opportunities that have come along. And here are some of the milestones in the program. So, so we started in my lab developing this now about 10 years ago, and 7 years ago now received approval from the New York State Department of Health. So any clinical test that's run in a clinical setting to report results back to patients, to use to guide clinical decisions, this all has to be done in a regulated environment. So this test happens to be available both in our research genomics core at MSK, as well as our clinical laboratory in the Department of Pathology, and all the data can be integrated and mined together. But for the patient reporting, it has to be done in this environment. And we actually obtained US Food and Drug Administration approval or authorization, technically in 2017, and have now sequenced over 50,000 patients. I'll describe that in more detail. Just coming back very briefly to the patient consent. So when we first launched MSK Impact, there were, I'd say about 90% of patients are consenting to an institutional protocol. And the reason for this is that we paid for this at the very beginning, largely with research funds or philanthropy or institutional funds. And if we're using research funds to pay for the testing, it has to be done on a research protocol. So we wrote a protocol that allowed us to not just perform the clinical test, but also utilize leftover material for more comprehensive, let's say, whole exome or whole genome sequencing, or leftover tissue for RNA extraction or other analysis, reanalysis of the raw data produced through the sequencing effort, collection of additional samples. So most patients are consenting to this broad institutional protocol, which was really valuable. But there were some patients for whom we were already doing molecular profiling using more targeted assays as standard care. So patients with lung cancer, for instance, were already getting molecular testing, and that was just a diagnostic test. It didn't require any consent. Here, we made the determination that every patient who has MSK Impact performed has to sign some consent because of the possibility, even if it's remote, of incidentally uncovering a germline variant associated with cancer or some other disease. Because it's a matched tumor and normal assay and we're sequencing the normal, there had to be some consent. So already that was sort of a disruption to the workflow in the thoracic oncology clinics, where lung cancer patients were routinely subjected to more targeted mutation profiling. So as an alternative to the research consent, which is a considerable amount of a patient's visit to go through all the elements of it, a much more abbreviated clinical consent was drafted. So all patients are consenting to something, just to document whether or not in the sort of instance that we might find a germline result, whether or not they want to be informed of that, acknowledging that we're not always looking for that, and that's a separate part of this program, but that if we found something in the germline, they had to be on record as to whether or not they wanted to be informed. So that was a simpler path for the treating oncologist, but much more limiting in the kind of research that we could do. So everyone is strongly urged to here to consent their patients to the broader institutional protocol, but there is a small subset who have not. Probably more detail than you need to know, but it's some of the it's indicative of the internal deliberations we've had just launching this program with the appropriate consenting mechanism. To actually generate the data that I'm going to present. So as of today, we've sequenced over 72,000 tumor normal pairs. This is all done within our molecular diagnostic service in the Department of Pathology. So this is a list of the faculty and clinical fellows and lab technologists and clinical bioinformaticians. We have a clinical bioinformatics team of over 26 people right now, just to support this large-scale data generation endeavor. Not to mention the oncologists consenting patients, or surgeons or interventional radiologists producing tissue, or anatomical pathologists making diagnoses in these cases. So it's a very large effort just within our center. I referred to this briefly earlier, but ONCOK is the resource we use, and it's a publicly available resource to annotate the clinical and biological significance of the variants that we identify. So there's a whole schema for, and this involves the input from over 100 research and clinical fellows and faculty at MSK at this point, but it's really been driven by a team on the computational side by Nikki Schultz and GHGA and Devyani Chakrabarty on the scientific side, to leverage the literature, databases, treatment guidelines, conference abstracts, any information we can to vet the clinical relevance of individual mutations, not just at the gene level, but at the alteration level and at the cancer type level. And these annotations are available for all studies in the CBio portal. I'll come back to the CBio portal, but some of you may be familiar. And the whole tiering scheme is shown here for the strongest evidence where it's an FDA recognized biomarker for an FDA approved drug, down to more investigational and preclinical data. And all of this is available in the reports, every report that we issue from our molecular diagnostic service. Just want to mention also that we had a recent milestone a couple of weeks ago. It's the ONCOK team, and sort of congratulate them. I know they're not on the call, but for establishing this resource, a website that you can browse. But also recently was the first somatic mutation knowledge base to receive FDA in the US FDA recognition for clinical decision making, and this was a big announcement a couple weeks ago. Looking at the ONCOK annotations across our whole cohort of cancer patients, and this really does span all types of cancer. If we consider down to the ONCOK level 3B, which is where there's compelling clinical evidence from clinical trials, or the level 1 and 2, which are the FDA approved in the standard care biomarkers, it's just over 50%. of all patients have at least one, what we'd consider to be actionable mutation, using a fairly conservative actual definition of what's actionable. These are as agreed upon by the clinical community at MSK to meaningfully affect the specific treatment decisions that they would make. But it does vary, as you can see on the right, pretty widely across different cancer types. So having done this, I can comment a little bit on all the benefits of establishing this population scale clinical sequencing initiative. And we're often asked, was it worth the investment, particularly by the administrative arm of the hospital? I think in many ways it has been, more than worth the investment. Obviously, at the individual patient level, as part of the diagnosis and patient care enterprise, it's allowed us to deliver precision diagnostics and enable oncologists to select the most appropriate therapies. It's really been the driver for our clinical trials program, and that's a big part of what MSK has built its research program on. In that, in the past, there may have been different clinical trials focused on individual cancer types, where each trial might enroll a patient, and then the patient would get screened for relevant genomic alterations that might, for a targeted therapy, suggest that they might benefit from the drug. But rather than having separate trials with each of which its own separate screening effort, basically created one large enterprise-wide screening effort that feeds patients into all of the different clinical trials. And once we have outcome data, drug response data, we have the baseline comprehensive genomic analysis to perform correlative studies. That's just for clinical trials. So in aggregate, we also now have 72,000 tumor normal sequencing data sets that allow us to do more broad biomarker discovery, pan-cancer types of studies, given that each of these patients also has at least some degree of clinical outcome or drug response data where we can perform integrated analyses. And just as a resource for the hundreds of basic researchers and translational researchers with groups here, it has served as a basis for identifying variants for functional characterization, or patients where additional biospecimens could be collected, or leftover DNA can be sequenced by whole exome or whole genome sequencing. And this resource itself has forced us to really examine the best ways of maximizing the benefit. It's been a big investment. We want that to benefit researchers both here at MSK as well as in the broader community. So from the very beginning, data sharing was a key principle of our program. We were committed to sharing data. We recognized that, unlike in the past, where a lot of the genomic data generated through our genomics core was from an investigator-initiated study where an individual lab or investigator was submitting samples or had gotten grant funds or was generating data that, even though it officially all belongs to the institution, was still unofficially their data and their prerogative as to whether they wanted to share it or not. This was all institutional. This was a resource that no one person or group or department owned, and we wanted everybody to benefit. So we've been using the CBio portal. For those of you involved in cancer genomics, I think many of you may be familiar with this tool. It was originally developed here at MSK by Nikki Schultz and his team to enable the sharing and visualization of data from the Cancer Genome Atlas studies. We have sort of co-opted it internally for sharing with our MSK faculty and actually every MSK employee the data and results from MSK Impact in real time. Every night it gets updated. It's all de-identified as a companion to the individual identified clinical reports that get issued. And in fact, the patient's electronic medical record now includes links out from the patient's chart directly to the corresponding page in the CBio portal, because for those of you who use the portal, it's a great way of visualizing an individual genomic record in the context of all other genomic studies. How often have individual mutations, for instance, been reported or identified in different types of cancer? So this is actually an internal version that I'm showing, dashboard of our 72,000 tumors from 58,000 patients. But the majority of these are also available externally, publicly as well. Just by emphasizing the data sharing and making this a priority for our program, we maybe underestimating, but are aware of over 500 papers published by MSK investigators now featuring the genomic results from MSK Impact that's in the CBio portal. And this spans all disciplines of medicine as well as basic research labs here as well. The external data sharing, as I mentioned, is largely carried out through the AACR Genie consortium. So for those of you who don't know, Genie is a project launched a few years ago by AACR under the leadership actually of Charles Sawyers, who's here at MSK, who had been a past president of AACR, who basically through the AACR convened, and for those of you not in cancer, it's the American Association for Cancer Research, convened a consortium of the large cancer centers actually around the world. Most are in the US, but some European and Asian centers are participating as well, to pool all of the results. All of these centers are doing some form of MSK Impact sequencing locally. It has their own data workflow. And it's using different panels, and there are different depths of coverage and different modes of sequencing only tumors or matched tumor normal pairs like we do. So it is kind of a hodgepodge, but it has broken down barriers and allowed us to better characterize rare types of cancer or rare alleles, rare mutations that benefit from larger numbers. So AACR Genie is a publicly available resource. If you go to cbioportal.org/Genie, the current release is about 120,000 tumors. And as you can see, we're the blue wedge. We've been the primary contributors, along with the Dana-Farber Cancer Institute in Boston, the red wedge, to this initiative. So, the basic timeline is that, and this is to give investigators at the individual centers a chance to benefit from the data generated for their own research. I believe all of the centers have exclusive access to their sequencing data for 6 months from the time it's generated. Then at 6 months, it's available to all of the other Genie consortium members. And then at 12 months, it's available to the entire community. So, there's about a 1-year lag between what's in our internal version and what's available to the community. But it really, I think, does demonstrate a prioritization for data sharing for the centers that have joined. So we've had to do a lot of thinking then about how to promote data sharing, but still establishing guardrails to avoid potential issues with data sharing. One of which being there's a risk with more people accessing the data that they might misinterpret the results and publish a paper that is not correct, or that they might undermine some larger initiative that an entire disease team has been investing in for a long period of time. So there are a number of different reviews focusing on the internal use of data, but these principles have recently been applied to our external data sharing as well. Some of which have been well established. So we have, like every center, an IRB, an Institutional Review Board, reviewing all research to make sure that patients' interests and patient privacy is protected and that they're compliant with all of the regulatory requirements. There is an Office of Technology Development, our Tech Transfer Office at MSK reviews research to make sure the business interests are protected. But this third one is something that I think has been more challenging and often overlooked, is protection of research interests. And this happens a little bit sporadically, but in some cases not at all. And that's more in line of what I was saying before. Is it appropriate for anyone to do any analysis and publish any result? Or are there risks in that? And I mentioned the risk of misinterpretation, or is it appropriate for a graduate student in a basic research lab who has access to this because the lab's affiliated with MSK to then publish the entire set of breast cancer patients without collaborating with or even informing the breast cancer disease team who consented the patients, collected samples, were really responsible for the whole data set in that respect, and can probably help enhance the quality of the research or correct any misinterpretation. So this is something that we've been spending a lot of time thinking of, and this form of data governance has become increasingly urgent, in part because not just genomic data, but clinical data, imaging data, other forms of patient data are growing exponentially and becoming increasingly accessible to all affiliated staff. And the institution itself is continuing to invest heavily in creating aggregated and harmonized institutional data sets. And often, when these discussions have come up, there's a feeling that, oh, well, if there's something that's really bad that's about to be published, the IRB will catch it. But because we're creating de-identified versions of these data sets, a lot of these projects aren't even subject to IRB review because there's no risk of patient re-identification, and those issues have become exempt from IRB review. So there are really no checks that exist. So in dealing with this, we've established, again, just for internal data sharing, a data and tissue usage committee that oversees the use of MSK Impact and related resources. I now chair this group, but we've drawn from many different clinical and research departments at the hospital to try to give as broad a sort of visibility for what research is happening with the MSK Impact data as possible. And what is this group, what is the purpose of this? Well, largely is to prevent redundant or competing research efforts and prevent misinterpretation of results by including collaborators of diverse expertise. Also, just ensuring proper attribution, recognition of contributions. People that have the data don't really necessarily know where it came from might not properly acknowledge the molecular diagnostics staff members and the clinical team members who were heavily involved in generating, analyzing, interpreting, reporting the results. And it's really to establish protections for those key stakeholders as well. So it's this balance between broad sharing, but ensuring that stakeholders' interests are protected as well. So I'm happy to discuss that in a little more detail. I wanted to use the last 10 minutes or so to just give you a sense of the breadth of follow-on analyses that we're generating. Because so far I've just been talking about in the presence or absence of individual mutations in individual tumors, which was the initial priority for establishing the MSK Impact Initiative. We were hoping that in the majority of patients, we might find at least one mutation that could theoretically be targetable by an available therapy. But the fact that we're sequencing both the tumor and the normal DNA has allowed us to significantly broaden the information that we're getting from the data. So there's a somatic mutations were specific to the tumor and not the normal. I'll come back to these other categories in a bit. What we currently report, as I mentioned, are many different classes. So mutations, indels, copy number alterations, gene fusions, tumor mutation burden, and microsatellite instability. But by making not just the results through the portal, but the raw data as well available to computational biologists at our center, we've been able collectively to expand on a number of additional analyses that right now are more in the research realm, but might eventually be validated for clinical use and reported through the results. And this includes genome-wide features like allele-specific copy number, loss of heterozygosity patterns, whole genome doubling, which we can infer from MSK Impact, other mutation signatures. Not all of them are, homologous recombination deficiency is a little bit aspirational because I think targeted panels aren't ideal for this, but we can detect LOH and other mutational signatures associated with high mutation rates. And the properties of individual mutations, like the zygosity of the mutation, the clonality of the mutation. So with this broader set of features and properties of mutations, we have a pan-cancer working group just internally to MSK working on expanding this resource. And a few years ago now, we published the first 10,000 cases that were sequenced by MSK Impact, could look at the frequency of mutations in different genes across different cancer types. But with a much larger set, we can be more granular and look across different subtypes and look at other features like whole genome doubling and tumor mutation burden. The ONCOK annotations are shown down the right, as well as the properties of these mutations like clonality and zygosity.
Mike,
so
you may want to close it within 5 minutes to allow for a few questions.
I'm happy to do that. Yeah, thanks. So I mentioned some of the features. Some of these are more in the research realm, like whole genome doubling. We can characterize this across different types of cancer, and almost all the time we see it, it's associated with a worse survival. Properties of individual mutations. So how do we do this? Well, this comes in part from using a tool developed here in our biostatistics department called Facets by Ranglai Shen and Venkat Session, where we can use the imbalanced SNP frequencies, target about 2,000 SNPs across the genome to identify allele-specific copy number genome-wide. But also because of the deep coverage of the assay, we have much more precision for identifying variant allele fractions that combined with allele-specific copy number allow us to infer the number of copies of a mutation and whether it's clonal or subclonal. And just to show a little bit of that, we can distinguish balanced mutations where they're heterozygous and 1 out of 2 copies of the genome, from all forms of imbalance where you can lose one of the copies or amplify one of the alleles, and for even the same mutation across different patients. So KRAS mutations in pancreatic cancer or KRAS mutations in colon cancer, the patients who have imbalanced KRAS mutation, either through an amplification of the mutant allele or loss of the wild type allele, shown in these red curves, have consistently worse survival than patients with balanced KRAS mutations. So this is important clinically. For germline variants, just very briefly, we're identifying cancer susceptibility alleles. This is through a process overseen by our molecular genetics, clinical genetics group and diagnostic molecular genetics group. This is a follow-on analyses where the consenting is via a 6-minute video that patients watch on an iPad in the oncology clinic, agreeing to this informed germline analysis for cancer susceptibility alleles. We've, through this mechanism, identified germline analysis in about 23,000 cases, and 17% of patients across cancer have at least one pathogenic germline variant. And finally, clonal hematopoiesis. Some of you may be familiar. These are somatic mutations in blood cells or hematopoietic stem cells, not tumor cells, that can lead to a clonal expansion within the blood. And many of these mutations are in the same genes that are mutated in leukemias and other hematological malignancies, and shown that these mutations can be precursors to hematologic malignancies as well. So we have this resource now of clonal hematopoiesis calls for 50,000 patients. Overall, about 25 to 30% of all patients exhibit some form of clonal hematopoiesis. We know this is a strongly age-associated phenomenon, and that's partly why the rate is relatively high, because the patients with cancer at MSK tend to be on the older side as a whole. But also it's the deep coverage sequencing of blood that I think allows us greater detection sensitivity. And as I said, the patients with CH are at significantly increased risk of a hematological malignancy. And this prompted us as a center to create the first ever clonal hematopoiesis clinic for patients who are having these incidental findings of clonal hematopoiesis reported. And this has been a vehicle also for collecting additional biospecimens from these patients over time as they're being monitored for secondary malignancies. So I think I will maybe end in, if I can take a minute more, because it relates to a new resource that I wanted to share some of the research that we're doing with all this data, where we have this unique opportunity now to examine how germline variants influence somatic phenotypes by looking at the interplay and what's happening with germline variants in the resulting tumors. And is the germline variant retained? Is the second allele lost through a second mutation or loss of heterozygosity producing biallelic inactivation? This has been an effort that actually a large study is hopefully published within the next few weeks. It's in press at Nature Genetics right now, with a resource of not just the prevalence of variants by different cancer types, which we can identify fairly granularly, looking across different cancer susceptibility genes with pathogenic alleles across different types of cancer, where we find 5% of patients having at least one what we consider a high penetrance germline variant. But what's unique here is we can layer on top of this the rates of biallelic inactivation in the corresponding tumors, which can help us better identify the factors that mediate biallelic inactivation, which cancer types are significantly associated with biallelic inactivation, so loss of the second copy of a tumor suppressor gene, which can also help identify, so we sort of characterize the determinants of biallelic inactivation by the penetrance of the variant, by the gene where it arises, by the specific variant and the specific cancer type. And this is, more research, I guess, we described associated with this in our manuscript. But the reason I wanted to end here is to introduce this new website that will be open to the public. It's live now, but will be sort of officially launched with the paper. It's called signaldb.org, which summarizes this integrated genomic, integrated somatic and germline data in a cohort-wide way that prevents re-identification of patients, but includes not just prevalence, but rates of biallelic inactivation for different genes. So at the gene level, which gene is this? BRCA1, can see all the somatic and germline variants that we found, and for each germline variant that's recurrent across the cohort, how often is it associated with biallelic inactivation or a second hit? And then at the individual variant level, also the rate at which it's observed across different types of cancer. So I guess for the sake of time, probably better to end and take questions rather than iterate through the summary slide. I didn't have time to talk about cell-free DNA profiling, but there's a companion test called MSK Access that we've launched a couple years ago to complement MSK Impact for cell-free DNA analysis of patients with metastatic cancer. So I will just before I stop sharing, let me just jump to the acknowledgements. I already mentioned the molecular diagnostic service, but also members of my lab and our Center for Molecular Oncology and MSK that have been instrumental in carrying a lot of this research forward. So thanks very much. I'm happy to stay on for questions either in live or on the chat.
Thank you, Mike.